Dear students, in today's lecture we will discuss about haploid culture, as the name indicating that in this type of culturing only one parent will be involved. Either it will be a male parent or it will be a female parent. If it is a male parent, then in case of that it will be known as androgenesis, and if there is a female parent, then it will be known as the gynogenesis. Haploid culture is basically known to be the culture that includes anther and pollen from the male parent and ovary or ovule of the plant from the female plant. And this is basically the process in which you make the sporophyte with the help of gametophyte, and in this case only one set of chromosome is involved. Either it is the chromosome from the anther or pollen of male flower, or this can be from the ovary or ovule of the female flower. So this process is basically very effective for the detection of recessive mutation. As I have discussed, told you in the beginning, that there are two haploid culturing methods. One is the androgenesis and other one is gynogenesis. Androgenesis, as the name indicate, that this is prepared by the male gametophyte, and there are two methods to using these two methods you can go for the androgenesis. Here is the brief overview of the process, or pictorial view of the process, here, in which you can use the anther for culturing the for the growth or development of a plantlet. Anther culture is basically very simple culture, and in this no specific conditions are required. This process was originally developed by Guha and Maheshwari in 1964 and 1966. In this technique, we basically excise the closed flower buds which have anthers that contains uninucleate microspores. At this stage, microspores are basically the more suitable for the induction of androgenesis. The excised flower buds are obviously surface-sterilised, and then anthers are removed. Absolute care is taken during the process of anther removal to avoid any kind of injury, because if there is injury, there will be no androgenesis. Then, after the removal of the anthers, the cultures are incubated under suitable conditions, and depending upon the plant species, that which type of species you are using for the anther culture, it takes 3 to 8 weeks for pollen plantlet to regenerate from the anthers. After a month of incubation, the microspores either develop into embryos or calli emerging through their anther lobes. Subsequently, these calli can be transferred to regenerate mediums, and some pollen grains develops into embryo while others develop from into the calli. So this is also a flow chart which is indicating that you are using the donor plantlet section, and after the pre-treatment and surface sterilization, that will be used for the process of regeneration of the plantlet. Another method is the pollen culture, that can be used for that can be genetically modified before culturing, and this is basically genetic modification is done for improving the productivity. And exogenous treatment is effectively applied, and best method for the study of cellular and subcellular changes. In this case, flower buds are removed from donor plants which is older than 6 weeks, and then bud length was 2 to 4 millimeter, and that only could range to produce the embryos. The buds were placed in test tubes, and then surface sterilization is done, and then these microspores are suspended in the petri dishes, and then dishes were wrapped in double layer of parafilm. Every test was prepared at least 3 times, were repeated and 3 times, and incubated in dark medium for almost 2 to 3 weeks. So general procedure for the anther culture and polar culture is, first of all you have to collect the medium, and then you have to disinfect anthers or pollens, and after the material flower, either you are using the flower male bud or the female bud. From that you have to excise the pollens or anthers, then you need to culture, then microspore development analysis and handling of haploid plantlet is the next or the last method, last step of the androgenesis. The factors which affect basically the process of androgenesis, it includes first of all the condition or a condition of donor plant, that what kind of donor plant you are using and what is its mental, its health state. Then either it is infected with any algae, virus, bacteria or any other, or it is a healthy plant or a healthy flower. Then stage of the microspore development, either it has started any development or no. Then pre-treatment, either you are treating that with the temperature and light for the callus culture induction, and also the media in which you are growing these types of androgen for the androgenesis. This is basically the floral bud of asparagus. This figure is basically indicating that how the process of androgenesis has been done in the asparagus plant and the asparagus flower bud.